physiology for 3 years before entering the PhD program in integrated pathobiology at the University of California, Davis. Mauricio obtained his PhD degree last July. Congratulations.
Thank you.
As a postdoctoral scholar, Dr. Navarro's research focused on diseases produced by Clostridium, which we will hear more about today.
Well, hello everyone, and thank you for the introduction and for the opportunity to present here today. This presentation is based on some of the topics of my PhD dissertation about Clostridium perfringens in the context of intestinal disease and intestinal pathogenesis using animal models for the human condition. And also the search for potential therapeutics against this pathogen. This is where my hometown is located, in the south of the world, almost falling off the map, but we're still there. And this is the classic view of the city. Valdivia is mainly known for being the epicenter of the largest earthquake in the history of mankind, with 9.5 degrees in 1960. Anyway, the virulence of C. perfringens is mainly due to the production of several protein toxins, and the presence of the genes that encodes for these toxins allows a classification system, this updated classification system, that divides C. perfringens into 7 types, from A through G. And this presentation is focused on type F, which are those strains that produce only alpha toxin and Clostridium perfringens enterotoxin, or CPE. CPE is involved in several cases of food poisoning in humans, and in some cases of antibiotic-associated diarrhea. Sometimes, and under specific conditions, CPE can be lethal. The combination of CPE plus constipation, for example, allows the toxin to be in contact with the intestinal mucosa for a longer time, inducing not only extremely severe necrotizing colitis, this is a human patient, but also death due to the absorption of the toxin and its action on distant organ, a phenomenon that is known as enterotoxemia. CPE is unique in that it is only produced during sporulation. As the spore is forming, the cytoplasm of C. perfringens accumulates many copies of this toxin, which is then released when the spore is finally formed. CPE is a pore-forming toxin that allows the entry of calcium into enterocytes, inducing caspase-3-mediated apoptosis when cells are treated with low doses of CPE, or necrosis in the presence of high CPE doses. This pathogenesis, however, was proposed using cell cultures in vitro, but the real mechanism in vivo have not been explored. And considering a potential role of caspase-3 activation in vitro, the first question of this research project was, well, what is the actual contribution of caspase-3 to CPE-mediated disease in vivo? To answer this question, we developed a small intestinal loop model in mice. This basically consists in performing 2 ligations in the small intestine, under anesthesia, obviously, and then the purified and bioactive toxin is injected into the loop. We put the intestine back into the abdominal cavity, we close the abdominal wall, and let the toxin incubate. We first tested for increasing CPE concentrations for up to 2 hours. All these concentrations are physiologically relevant, meaning that they can be found in the stool of patients with CPE-associated diseases. And after the 2 hours, we look at the histology. And as you can see to the left, we have representative images of those treatments and histologic and semi-objective quantification of the histological damage. And as you can see, the intensity of this damage was dose-dependent, with the highest CPE concentration using the most severe epithelial desquamation, virus planting, and fusion of the villi. Not only the intensity of damage was dose-dependent, but also the activation of caspase-3, as measured by immunofluorescence here in red. And to this point, we already had a different result from those in vitro studies, in which they suggested that a high CPE dose, with a high CPE dose, apoptosis was not important, and cells died mainly by necrosis. So Oh, and this is just a confirmation of the previous result, in which we also measured caspase activation by colorimetric assay, and again, the same dose-dependent activation, with 100 micrograms of CPE inducing most significant activation. So considering that this caspase activation was dose-dependent, as it is the intensity of damage, we thought that maybe by blocking caspase activation, we could prevent those intestinal loops from the histological damage. So we created additional intestinal loops incubated with CPE, but this time in the presence of a pancreas-space inhibitor for up to 2 hours. And the same procedure extended to 4 hours, which is our enterotoxemia model for CPE. First of all, the presence of this inhibitor was very efficient in blocking most of the caspase activation at different time points. When we compare these values of activation against those loops receiving CPE only, both by colorimetric assay and immunofluorescence. However, the presence of this caspase inhibitor wasn't able to protect those intestinal loops from intestinal damage, as you can see in this plot and in these images. So this was the conclusion until this point. This inhibitor works very well in preventing caspase activation, but it doesn't protect from histological damage. This treatment didn't protect mice from enterotoxemia either. As you can see, this proportion of mice that survived until the end of the experiment there was no difference between the presence of the absence of the caspase inhibitor. So considering that this inhibitor didn't protect from histological damage or enterotoxemia, obviously it doesn't represent a good candidate to treat CPE-associated disease. However, a recent study done by a research group found that this drug, mepacrine, was able to interfere with the formation of CPE pores and the activity of those pores already present in the plasma membrane, which is the basis for the next question of this study. Does mepacrine interfere with CPE action in vivo? So what's mepacrine? This is an acridine derivative drug. It's actually an organic compound which has this intense yellow color. The mechanism of action of mepacrine is not really known, but it was widely used during the 1930s to treat and prevent cases of malaria in humans during the Second War. And more recently, it has been used as an alternative to treat intestinal protozoal infections, particularly against Giardia. So the fact that it is already used to treat intestinal diseases was kind of the rationale to investigate this drug against CPE. So we use the same intestinal loop model, this time injecting the toxin in the presence of a high or a low dose of mepacrine, the toxin only or the drug only. We then look at histology and took serum samples from different purposes that I'm going to comment in a few seconds. The first observation with this treatment is the protection against CPE-induced enterotoxemia. Again, when we see the proportion of mice that died until the end of the experiment with the toxin only, compared with the mice that survived in the presence of mepacrine, it was significantly improved. A low mepacrine dose, however, didn't protect from this enterotoxemic death. When we look at histology, the presence of either mepacrine concentration was able to reduce the intestinal damage. This protection is not complete since there is still some epithelial desquamation, as you can see in this figure, but the overall intensity of damage was significantly reduced. So how is mepacrine protecting these mice? We already know that the formation of CPE pores is essential for CPE to induce cell death. So we thought that maybe mepacrine was interfering with the spore formation in the intestinal loop, as it does in vitro. However, when we look for those pores in these intestinal loops by Western blot, there was actually no difference. So even in the presence of mepacrine, those pores are still present. Perhaps mepacrine was reducing the entry of CPE from the loop into the blood, For that reason, we measure the levels of the toxin in the serum in the different treatments. And the reduction in the absorption of CPE was true for some of the mice, but the overall variation among animals says that there was actually no difference. So this is not really explaining how mepacrin is protecting. So the question remained, and in this paper of 2011, they found that the incubation of CPE in chalup was actually correlated with an increase in the levels of potassium in the blood. So it was proposed that what actually kills the animal is a state of severe hyperpotassemia induced by CPE and the consequent cardiac arrest. So with that information, we measured the levels of potassium in our mice, and we found that those receiving a high mepacrin dose had potassium levels similar to those receiving the drug or the buffer only. This plot actually correlates well with this survival curve that I showed before, in which a high mepacrin dose protects from CPE-induced enterotoxemia. So the main conclusions of this work, first of all, CPE induces caspase activation in vivo. We can block this activation, but it's not essential to cause either intestinal damage or enterotoxemia, so it doesn't represent a good pharmacological target. And mepacrin might be considered as a potential therapeutic against this disease because it protects from histological damage and enterotoxemia. Finally, I'd like to acknowledge all the people involved in this project, particularly to my major professor, Dr. Francisco Salas, UC Davis, and Dr. Bruce McLean at the University of Pittsburgh in Pennsylvania, and as well as all my funding sources. Thank you very much for your attention.
Questions for Dr. Navarro?
Will you make a comment about the possible or non-role of CPE in animal disease?
Well, that's a very good question. And
could you repeat the question?
Oh, yeah. Dr. Salas is asking about a potential role of CPE in animal disease. And the role of CPE in animal disease is kind of controversial because it can be isolated. We can isolate C. perfringens strains that are CPE positive in healthy animals or in animals with disease. So what's the meaning of that? We don't really know. We know that CPE affects the intestinal mucosa when we test it directly. And as I mentioned before, CPE is only produced during sporulation, and the mechanism of sporulation in animals in natural conditions has not been explored, so we don't really know if there is an actual role in animals. And actually, the disease in humans is associated with an ingestion of large amounts of C. perfringens. An ingestion of C. perfringens, since that doesn't happen in animals.
So with the mepacrin, does it affect, or has it been used pharmacologically in animals, to your knowledge, other than obviously the FDA testing that had to have occurred at some point?
Correct, that's a good question. I don't think it has been used in animals, actually. And thinking about CPE, maybe it's not that relevant because we actually don't know if CPE is doing something in animals. But considering that CPE is an important pathogen virulence factor for humans, that was the key of this research.
We treat Giardia malaria in animals. Was it been used for those purposes at all?
I don't actually know about that.
I have a question. Is the mechanism of action related to potassium control for those diseases as well, for Giardia and the other intestinal diseases for mepacrin use?
Oh yeah, I think the question was if mepacrin has something to do with the levels of potassium
when it's used
for other diseases. Okay, I don't think so because those intestinal diseases produced by protozoa are local, meaning that they act only in the intestine. So they don't have an effect in the level of potassium in the blood. The thing with CPE is that it can be absorbed and affect distant organs.
Have you ever used antagonistic loops inside the same animal to see the effect? In other words, the loop is treated and not see if it would ameliorate the lesion.
So the question is, if we have
To use antagonizing loops within the same animal.
in the same animals different loops, yes, and actually that's something that we do in rabbits because well, it's a longer GI tract, and the problem with that is we cannot evaluate enterotoxemia, but the local effect of the toxin in one loop or buffering only in the other loop.
There's a lot of clinical disease in the folks.
We haven't tried that as a clinical condition.
Any other questions for Dr. Navarro? Thank you very much, Dr.
Thank you.
Thank you all